Hello to everybody and welcome to all. I am Dr. M. Arumugham, Assistant Professor and Head, PG and Research Department of Botany, JJ College of Arts and Science, Autonomous, Pudukkottai, Tamil Nadu. And today the topic of my presentation is about the cellular organelles. First, I would like to say the structure of cell. The structure of the cell, when you go under the microscope, we would come across the three distinguished features. One is plasma membrane, another one is nucleus, and third one is cytoplasm. All the activities inside the cell and interactions of the cell with its environment are possible due to these features alone. And first the plasma membrane, and next one is nucleus and cytoplasm. Then the cytoplasm also consists of, one is a cytosol and another one is a cellular organelle. And the cellular organelle is concerned right from the endoplasmic reticulum, Golgi body, lysosomes, vacuoles, mitochondria, plastids, centrosomes, and also cytoskeletons. And first I would start with the organelle plasma membrane. It is extremely delicate, a very thin, elastic and living. Also it is a special character is semi-permeable membrane. It made up of two layers of lipid molecules in which protein molecules will be floating. The thickness of this plasma membrane varies from 75 to 110 Armstrong. The plasma membrane can be observed under the electron microscope only. The functions of the plasma membrane is concerned, it maintains shape and size of the cell, and it protects the internal content of the cell, and also it regulates entry and exit of substances in and out of the cell, and it maintains the hemostasis also. And another one is cell wall. The first of all, the cell wall is non-living and outermost covering of the cell, especially in plants and the bacteria. It can be tough, rigid, as well as sometimes very flexible. It made up of cellulose, hemicellulose, and also pectin. And the cell wall may be a thin or thick, multi-layered structures, but multi-layered structures. The thickness about the cell wall varies from 52,000 Armstrongs. The main functions of the cell wall it is providing a definite shape, strength, as well as the rigidity of and living things. It prevents drying up, that is called desiccation of the cell, and also helps in controlling the cell expansion. It protects the cell from the external pathogens. And third one is very important organelle called nucleus. The nucleus is dense spherical body located near the center of the cell. The diameter varies from the 10 to 20 micrometer. Also it present in all the cells except two cells. One is red blood cells, another one is sieve tube cells. The nucleus is well developed in plant cells and the meantime in animal cells also. Underdeveloped in the bacterial as well as some blue-green algae, that is cyanobacteria, and also in the mycoplasma like cells. Most of the cells are uninucleates, but only in the skeletal cells, that is skeletal muscular cells, have the more than two nucleus. And the nucleus has double layered covering called nuclear membrane. So the nucleus is covered with a double membrane, that is called nuclear membrane, the nuclear membrane has a pores of diameter about 80 to 100 nanometer. The colorless, dense sap presence inside the nucleus known as nucleoplasm. Then the nucleoplasm contains the round shaped nucleolus in the center of the region, and it's covered with some net like structure is called chromatid fibers. The fibers are composed of DNA, that is deoxyribonucleic acids, and histone proteins, and these fibers condensed to form the chromosomes during the cell division. And the chromosomes contains the stretches of DNA called genes. This is a gene. This is the gene. Then this is called a DNA. This is called a DNA structure. And gene transfer hereditary information from the one generation to next generation. So the major functions of the nucleus, it controls all the cell activities because it is the power, it has a controlling capacity like metabolism, protein synthesis, growth and developments, as well as the cell divisions. The nucleolus will synthesize the ribonucleic acids, that is RNA, to constitute the ribosomes. Then it stores hereditary information in the genes to control all the organisms. And cytoplasm is occupy the maximum place in the cell. It is the place of intermediate between nuclear membrane to cellular membrane. Then it is a jelly like structure formed by maximum of 80% of water. The present between the plasma membrane and the nuclear membrane. It contains a clear liquid portion called cytosol. It varies in the particles. And the particles are the proteins, carbohydrates, nucleic acids, lipids, and inorganic ions also. It also contains many organelles with distinguished structures and also the specific functions. Some of these organelles are visible only in under the electron microscope invisibilations. The granular and dense in animal cells and thin in the plant cells. Then endoplasmic reticulum, it is net-like structure. The net-like structure of tubular vesicular structures which are interconnected with one another. Some parts are connected to the nuclear membrane while in other connected with the cellular membrane. So these two types, one is the endoplasmic reticulums are the two types. One is smooth and another one is rough. The smooth is lacking of ribosome and the rough endoplasmic reticulum is filled with or attached with ribosomes. The major functions of the endoplasmic reticulum is: one, it gives the internal support to the cytoplasm. The rough endoplasmic reticulum synthesizes the secretory proteins and membrane proteins. Then smooth endoplasmic reticulum synthesizes the lipids for the cellular membrane. In liver cells, smooth endoplasmic reticulum detoxify the drugs as well as the toxins. And in muscular cells, the smooth endoplasmic reticulum stores the calcium ions also. And this is Golgi body. This is a Golgi body. And the Golgi body is discovered by Camille Golgi. That is formed by stalks of 5 to 8 membranous sacs. The sacs are usually flattened and called as a cisternae. That it has two ends. One is cis phase as well as called trans phase. The cis phase is situated near the endoplasmic reticulum and trans phase is situated in the cellular membrane. The main functions of this Golgi body is: one is it modifies, sorts and packs of material synthesized in a cell. it delivers synthesized material to various targets inside the cell and outside the cell. It produces the vacuoles and secretory vesicles, and also it forms the plasma membrane and lysosomes. And this image indicates that how the Golgi body works out. From the endoplasmic, rough endoplasmic reticulum, vesicles will come out. From the vesicles, it attached with another plasma membrane. Then it will give say the new Golgi body. Then next, this is the lysosomes. Small spherical single membrane sac is called lysosomes. It found throughout the cytoplasm. It is scattered in the cytoplasm. It filled with hydrolytic enzymes. Then only it can autolysis some dead particles. Then it in occur most of the animal cells and few types of the plant cells. The main functions of the lysosome is concerned. It helps in the digestion of large molecules. It protects cell by destroying foreign invaders like bacteria and also the viruses. Degradation worm out the organelles. In dead cells perform the autolysis also. Then the vacuoles. This is the vacuole. This is a vacuole. The single membrane sac filled with a liquid or the sap, water, sugar and also ions. In animal cells, the vacuoles are temporary, very small in size and few in numbers. But in the case of plant cells, vacuoles are very in large size but more in number. May be a contractile or the non-contractiles. The main functions of the vacuole is it stores various substances including the waste products within the cell. It maintains the osmotic pressure of the cell, and also the store food particles in amoeba cells also. Then it provides turgidity and rigidity to the plant cell. And it is important one is mitochondria. The mitochondria is very small rod shaped organelle bounded by a two membrane, inner as well as outer. The outer membrane is smooth and enclosed with the contents of mitochondria. The inner membrane is folded in the form of self-like invaded projections called cisternae. This inner cavity is filled with matrix that is contains the many coenzymes, that is F1 particles. Then contains also the DNA, the individual own DNA consisting of mitochondria are responsible for the many enzymatic factors. The function of the mitochondria is concerned, it synthesizes the energy and also rich compound of ATP molecules. The ATP molecules will provide energy for the vital activities to the all living things. Then plastids. The plastids are double layered, that is a double membrane bounded. Then organelle found only in a plant cell or in some bacterial cell or the blue-green algae cells. These are the responsible for the activities related to making and storing of food. These they often contain the different types of pigments. They can change their color in the cell. So the plastids are the chromoplast, chloroplast and leucoplast. Then chromoplast having the different colors, and the chloroplast having the only green in color. Leucoplast is called don't have any color. Then leucoplast is the elaioplast and ameloplast as well as the proteinoplast. Then chromoplast. The chromoplasts are the plastids that produces and store the pigments. They are the responsible for the different colors found in the leaves, fruits, flowers as well as the vegetables. For an example, the carrot expressing their own color by the pigment is called carotenes, and the mango having the yellow in color by the expression of xanthophyll pigments, and then tomato it having the red in color means it expressing the pigment is called lycopene. Then the leucoplasts are the colorless plastids and store some food. They are found in storage organelles like fruits, tubers as well as the seeds. The potato tubers store some food that is called starch. The starch also leucoplast. Then maize grains is called food, it is a protein, and castor seeds it stores in the oil. Next one is the chloroplast. The chloroplast is a distinguished character of the plant cell, and another one is blue-green algae. It is a double membrane bound organelle bound mainly in the plant cell. It is usually spherical or discoid in shape, shows two distinct regions, grana and stroma. Another one is grana or stalked of thylakoids. It is called membrane bounded flattened discs. Another one is called thylakoid, containing the chlorophyll molecule which are responsible for the photosynthesis. And another one is stroma is colorless, dense fluid. Then main functions of the chloroplast is, first it converts the light energy into chemical energy to form a food molecules in the plant cell. It provides the green color to the leaves, stems as well as the vegetables. And another one is centrosomes. The centrosome is the membrane bound organelle present in the nucleus. Consists of two structures called centrioles. Then centrioles are the hollow cylindrical structures made up of microtubules. Then centrioles are arranged at in the angles to each and other. The main functions of the centrosome is concerned, it forms spindle fibers which helps in the movement of chromosomes during the cell division. It helps in the formation of cilia as well as the flagella. And another one is cytoskeletons. The cytoskeletons is formed by the microtubules and microfilaments. The microtubules are hollow tubes that makes up the protein called tubulin. The microfilaments are the rod-shaped thin filaments made up of protein called actins. The main functions of the cytoskeletons determine the shape as well as structure of the plant cell. It gives a structure strength to the plant cell, that is plant cell. The responsible for the cellular movements also. This is the major differences between the prokaryotic cell as well as the eukaryotic cell. First one is, in a prokaryotic cell, nucleus is underdeveloped, and eukaryotic cell is, the nucleus is well developed. So another one is, then only one chromosome present in the prokaryotic cell, but in a eukaryotic cell more than one chromosomes will be present. And in eukaryotic cell, membrane bound organelle are present, and third one is membrane bound organelle are present. Then here the sizes from the ranges from the prokaryotic cell is 0.5 to 5 millimeter, but eukaryotic cell 5 to 100 micrometer. Then examples, the bacteria and blue-green algae in eukaryotic cells, all the other cells. Then animal cells, plant cell differentiation is concerned. First is, in the animal cells generally it is a very small in size, but in the plant cells are in a large in size. In animal cells, the cell wall is absent, but in plant cell, cell wall is present. In animal cell, plastids are the present. In plant cell, plastids are the present, but in animal cell, the plastids are absent. The vacuoles are smaller in size and less in number, but in the plant cells, vacuoles are larger in size and more in number. In animal cells, centrioles are present, but in the plant cell, centrioles are the absent. Thank you one and all for the listening to my presentation. Thank you one and all.